Good morning, children. Again, welcome to biology class. Today, we'll continue again chapter Principles of Inheritance and Variations. So, children, we have already studied about the heredity, variations, blending characters. So, today we are going to start the Mendel's Principles of Inheritance. You know, children, that Gregor Johann Mendel, he was an Austrian monk, and he was the first person who explains the mechanism of transmission of characters from parents to offspring generation after generation. So he is known as the father of genetics. He conducted his historic crossbreeding experiments with garden pea for about 9 years. But his work remained unnoticed for about 34 years because the result, it was published in obscure journal, and the scientists, they were already preoccupied with the controversy which was thrown by Darwin's theory of origin of species. As the scientists of that period, they were also ignorant about the cytological basis of heredity. Mendel used mathematical basis to explain the results of crossbreeding, and use of mathematics to explain biological phenomena, it was totally new to the scientist. Now again his work was rediscovered by 3 different scientists: Hugo de Vries from Holland, Karl Correns from Germany, and Erich von Schirmack from Austria. They were working independently on heredity in plants. So Correns, he raised the status of Mendel's 2 generalization to the level of laws of heredity, law of segregation, and law of independent assortment. Bateson also found that Mendel's laws apply to animals as well. So, to recognize Mendel's contribution, these scientists, they name these laws as Mendel's law. Now, Bateson, Punnett, and subsequent workers, they found that the universal application of Mendel's law. So, what was the reason behind the Mendel's success? It was the combination of luck, foresight, mathematical approach, scientific aptitude, which were contributed to the success of Mendel's experiment. His method of working was also different. He studied inheritance of one character at a time, whereas other scientists, they had already considered the organism as a whole. Mendel carried out the experiments up to 2nd and 3rd generation. He also maintained the statistical records of all the experiments. Crossbreeding, it was done between the parents of pure lines, and the pure lines of parents, they were ascertained by a certain series of self-crossing tests between progeny of each successive generation. So why Mendel selected the pea plant? He selected the garden pea plant as experimental plant because it was an annual plant. It has short life cycle, and Mendel was able to study several generations of the plant within a short period. Its flowers, they are also complete and predominantly self-pollinating. Now, because of this self-pollination, the plants, they are homozygous, or we can say the pure lines of pea plant were available. Pea plant, it has a number of easily detectable contrasting characters which are related as dominant and recessive. And this pea plant, it also produces large number of seeds in one generation. So selection of traits, crossing techniques, and rest of the topics will cover in our next class. Till then, thank you and have a nice day.